All right, so this is page B1. Now, the first thing I want to remind you of that you learned in biology is something called the cell theory. And the cell theory was proposed in 1838 by Schleiden, Schwann, and Virchow. First comment, I never test you on names of people or dates. That would be a history class. So we're not asking you to know the names of the individuals or the date. I always thought that these guys, Schleiden, Schwann, and Virchow, would make a great name for a law firm. You know, Schleiden, Schwann, and Virchow, attorneys at law, LLD. But anyhow, who are they? Schleiden was a zoologist. I'm sorry, he was a botanist. And everything that Schleiden would look at in the 1800s with a magnifying lens, any plant that he looked at, he found that when he looked at it, a leaf of a plant, it was made up of cells. Those of you who had biology, if you took the biology lab, you had to look at the leaves of something, a plant that lives in water called an elodea or elodea. And you actually saw it was made out of plant cells. And every part of a plant, he said, was made up of cells. Theodor Schwann was a zoologist. He studied animals. And every time he took a magnifying lens and looked at any part of an animal, he said it was made up of cells. Rudolf Virchow was a medical doctor. He was a pathologist, an expert in human disease. And every part of the human body that he examined with a magnifying lens, whether it was the skin or the blood or the liver or the muscles, he said they were made up of cells. So in the early 1800s, these three guys got together and they said, it looks like all plants are made up of cells. Looks like all the animals are made up of cells. Looks like humans are made up of cells. You know what? All living things must be made up of cells. That's called the cell theory. And not only did they say that the cell is the basic unit of life, but that, that even more amazingly, all living things begin life as a single microscopic cell. So it's hard to believe, but at one time you were one cell big. And even an elephant or a whale starts out one cell big. So that's basically the cell theory. Now, the next question we want to address is why are cells so small? Because you need a magnifying lens or a microscope to see a cell. Why can't a cell just keep growing bigger and bigger and bigger? Now, your first thought is, because that's the way it is. I know, but in science, we're trying to understand mechanisms and how things work. So the question is, how come cells will grow, and they do grow bigger, and at a certain point, they stop growing bigger, and they do one of two things. They either stop growing bigger, and they just stay that size, or they divide into two little cells, and those two little cells start growing bigger. So we want to talk about the limits in cell size and why it is that any organisms that are bigger than a single cell, like us, will be made up of lots of cells, because we're made up of about 60 trillion cells. Now, what I'd like to have you do is just, if you have the lecture outline, and if you don't, it's okay, you can just see this. This is page B2I. So you should have a page that looks a little bit like this. And all that it's showing on this page B2I, it's right after page B2, it's just showing the idea that a cell is growing bigger and bigger and bigger, and then what happened to the cell? It divided into two cells, exactly what we said typically happens. Now, before I try to explain why cells can only grow so large, I want to remind you of another concept that, again, you should have learned about in biology. So, and if you don't have a lecture outline, it's real simple. You just draw a circle, which represents a cell. And what I've indicated here, these are nutrients that go into the cell in order to keep the cell alive. And these are waste products that go out of the cell, because if these waste products were to accumulate within the cell, they would become toxic, poisonous, harmful to the cell if they accumulated within the cell. Now, of course, this outer surface of the cell is called the cell membrane. That forms the outer surface. So I'll remind you of some things you learned in a college biology class. Some of the nutrients that all living cells, all cells, need are oxygen and sugars, like glucose. And the reason why all cells, including, we'll focus on human cells, need oxygen and glucose is for a process called cellular respiration. In biology, you learn that cellular respiration is the name of the process by which organic compounds such as sugars are broken apart with oxygen for the purpose of producing what? What are they trying to produce by breaking apart sugars with oxygen? ATP, the magic name, ATP. Now, if you're thinking, my gosh, I don't even know what the hell that is, you're in the wrong class. You're in the wrong class. Okay? Go back to biology, retake it if you've had it, or take it for the first time. But ATP is the high-energy nucleotide, the gasoline that's used to power all living cells, and that's produced in a process called cellular respiration. Now, interestingly, as cells carry on cellular respiration, producing ATP, they generate a waste product called carbon dioxide. And this CO2 has to go out of the cell across that cell membrane. So these are nutrients that enter the cell through that cell membrane, and CO2 is a waste product that goes out of the cell across that cell membrane. Now, obviously, if the oxygen couldn't enter the cell fast enough, then the cell would die because it wouldn't be able to produce enough of this gasoline called ATP. If the carbon dioxide could not get out of the cell fast enough, then the buildup, the accumulation of carbon dioxide inside the cell would cause the cell to die. So clearly, these nutrients have to get into the cell through that cell membrane fast enough, and these waste products have to get out of the cell across that cell membrane fast enough. Other nutrients that all cells, including human cells, require in order to live are amino acids. You learned in biology that amino acids are the so-called building blocks needed to manufacture or synthesize proteins. Proteins are built out of amino acids. So these have to get across the cell membrane into the cell fast enough so it can manufacture the proteins that it requires. Interestingly, and you may or may not have learned this in a biology class, but not only do cells manufacture new proteins using amino acids, all cells, all human cells, break down old proteins. And they actually break apart old proteins, forming waste products called ammonia and urea. So ammonia and urea are actually waste products formed from the breakdown of old proteins. And they have to get out of the cell. Now, other nutrients required by human cells, living cells, in order to live are nucleotides. And in biology, you learn that they include like adenine, guanine, cytosine, thymine, uracil. And these nucleotides have to get across the cell membrane into the cell in order for the cell to manufacture DNA and RNA, these nucleic acids. Obviously, if these nucleotides can't enter the cell fast enough, the cell won't be able to manufacture the nucleic acids they require. Something, again, you may or may not have learned in biology, but we're mentioning it now, is that not only do cells manufacture new nucleic acids, but they break down old nucleic acids. And when they break apart nucleic acids, they form a waste product called uric acid. So uric acid is actually the name of a waste product formed from the breakdown of old nucleic acids. Now, other nutrients that have to get across that cell membrane so that the cell can stay alive are vitamins and minerals. In a biology class, you learned that vitamins and minerals are coenzymes. Most vitamins and minerals assist enzymes in catalyzing biochemical reactions. Vitamins are organic coenzymes and minerals are inorganic coenzymes. And of course, we know that all living things are mostly made out of water. We're mostly made out of water. We're about 60% water. And the cell is about 80% water. And water goes into the cell, across the cell membrane, and water can flow out of the cell across the cell membrane. You might even remember learning in biology that whenever water flows into a cell or out of a cell, that's called osmosis. So the water has to be able to go in, the water has to be able to go out. Okay, so in summarizing, various nutrients have to get across that cell membrane into the cell fast enough to allow it to stay alive. And various waste products have to flow out across that cell membrane so they don't accumulate in the cell, killing the cell. All right, now that we've reminded you of that, let's look at the diagram right above. Basically, the reason why, what we've discovered of why cells all stay small and they cannot grow beyond a certain size is the relationship or ratio between the outer surface area and its internal volume. And you should have learned about this in biology, but whether you did or not, we'll explain it right now. Now, there is a formula we can use that allows us to estimate the outer surface area of a three-dimensional cell, a ball, a sphere. And the surface area, the outer surface area of a ball-shaped cell is 4 pi r squared. Now, if you're thinking, 4 pi r squared, I remember from a long time ago something like the area of a circle. You might remember that the area of a circle is equal to pi r squared. Anybody remember that? And so the way that works, an area of a circle, is the radius is the distance from the center of the circle to the outer edge. So if you simply square the radius and multiply times pi, that will tell you the area of this circle, how much area this circle is. So if that's the area of a circle is pi r squared, then why did I say that the area of a cell is 4 pi r squared? Because we're not talking about a flat two-dimensional circle, we're talking about a ball-shaped spherical three-dimensional cell. And its outer surface is equal to 4 pi r squared. Now, the internal volume of a cell, the internal volume can be calculated. The internal volume is 4/3 pi r cubed. So if we wanted to calculate the volume of what's inside the cell, it's 4/3 pi r cubed. Now the bottom line is what's limiting how big a cell can grow is the ratio, the difference between the surface area and the volume. So to make the math really simple, we're really just comparing these two formulas. Since both formulas have pi in them, which is 3.14, let's just ignore it. We'll just ignore it. They both have that pi unit in it. The difference between the number 4 and the 4/3 isn't a big deal. Let's just ignore that. So now, you'll notice that the big difference between calculating the outer surface of a cell and its internal volume is the surface area is related to the square of the radius, that's the outer surface, but its internal volume is related to the cube of the radius. Now, let's see how this works. Let's imagine we've got a little cell, and this is the radius. The radius goes from the center of the cell to the outer edge. And let's say the radius is 1. Now, you might say 1 what? What are the units? The units really don't matter because we're really just comparing surface area to volume. Whether we say it's 1 millimeter or 1 micrometer, it doesn't matter. Now, if the radius is 1, let's estimate its outer surface and the internal volume of this cell with a radius of 1. So, how do we estimate the outer surface? It's approximately the square of the radius. So if the radius is 1, what's 1 squared? 1 times 1 is 1. Now that's the outer surface area, right? We've just estimated its outer surface. All right, now let's estimate its internal volume. So the internal volume is approximately the cube of the radius. If the radius is 1, what's 1 cubed? 1. 1 times 1 times 1. So you'll notice that when a cell has a radius of 1 micrometer, 1 millimeter, the volume and area have the same basic size. Now let's imagine the cell grows larger. So now the radius is 2. So now, if the radius is 2, let's estimate the outer surface area. So what did we say this? How do we estimate the outer surface? It's the radius squared. So if this radius is 2, what's 2 squared? 4. 2 times 2 is 4. Let's estimate the internal volume. The volume is the radius cubed. So if the radius is 2, what's 2 cubed? 8. Some of you might say, I don't even know what he's talking about. What does 2 cubed mean? 2 times 2 times 2. 2 times itself 3 times. So 2 times 2 times 2 is 8. Can everybody see now how the volume has become much bigger number than the outer surface area? Let's try this one more time. Let's imagine the cell enlarges now even more. So its radius is 4. Now, with the cell with a radius of 4, let's estimate its outer surface and then its internal volume. So the outer surface is going to be the radius, which is 4 squared. What's 4 squared? 16. 4 times 4 is 16. Let's now estimate the internal volume of what's inside the cell. That's going to be 4 cubed. That's 4 times 4 times 4, which is 64. Can everybody see that as this cell is getting bigger, its volume is increased, its internal volume is increasing at a faster rate than its outer surface? The internal volume of what's inside the cell is increasing with the cube of the radius, but its outer surface is only increasing with the square of the radius. It's simply geometrical. It's mathematics. So here's, you'd say, well, I understand, I follow what you're saying, but I don't get it. What's the point? The point is that as this cell is getting bigger, it needs more and more nutrients to get into it, to stay alive. But its surface area isn't keeping up with its internal volume. Now, how do nutrients get into the cell? By moving through the surface. How do the waste products get out of the cell? By moving across that outer surface. So as a cell grows larger and larger, the nutrients can't get into the cell fast enough to allow it to keep living. And the waste products can't get out of the cell fast enough without accumulating and killing the cell. So when a cell grows beyond a certain size, it dies. So literally all cells are going to be microscopically small because when they grow beyond a certain size, the nutrients can't get into the cell fast enough and the waste products can't get out of the cell fast enough and the cell dies. So that basically, mathematically, is why a cell will grow, and when it reaches a certain size, it either stops growing and just stays that size, because if it grows any bigger it will die, or it divides and becomes 2 cells. And then when those 2 cells grow, they reach a certain size. If they grow beyond that, they'll die. So then what will they do? They will divide. So the way we grow, the way we develop from a single-celled zygote, fertilized egg, is those cells enlarged. When they reached when that cell reached a certain size, it divided, and we became 2-celled. Those grew, and when they reached a certain size, they divided. We became 4-celled. Those grew in size and reached a certain size, and we became 8-celled, 16, 32, 64, 128 cells, 256, 512, 1096. And that's why cells, that's how we increase the number of cells. So everybody follow. Understand what is the limiting factor of why cells stay small, and why, if we're going to be a big organism, bigger than a single microscopic cell, we're going to have to be made out of lots of microscopic cells. We can't just be made up of a big giant cell. It's really simply the ratio, the relationship between the relative difference between the outer surface area and the internal volume. Okay, now that we've reviewed that concept, back on page B1. Another thing on page B1, that you covered in biology is the types of cells. And you learned in biology that when we examine the cells of different living things, they fall into 2 broad categories: prokaryotic cells and eukaryotic cells. Now, what this basically means, first, the root, karyo, means, anybody know what karyo means? Nucleus. Now that I've told you what it means, you need to know that. So the word karyo means nucleus. Make sure you know that. That root is going to keep reappearing. So a prokaryotic cell is a cell without a nucleus. Pro means before. They evolved before there was a nucleus. So they lack a nucleus, and in fact, not only do these cells lack a nucleus, they lack also most other internal organelles, and they're very, very small cells. What's an example of a prokaryotic cell? Bacterial cells. They're very small and very simple. Most bacterial cells are 1 or 2 micrometers in diameter. Very, very small. When you take microbiology, for those of you who need it, you're going to spend most of the semester in microbiology talking about these different types of prokaryotic bacterial cells that cause various types of infectious diseases in us and in other living things. Now, the cells of most other living things are said to be eukaryotic. Karyo means nucleus. Eu is a Latin root that means true. They are true nucleated cells. Not only do they possess a nucleus, but they have many, many other organelles, little organs inside the cell. They are also much, much larger cells than bacterial cells. If we were to relatively compare the size of a human cell, which has a nucleus, with a bacterial cell, it would be like this. A typical human cell is about 100 micrometers in diameter. A typical bacterial cell is 1 micrometer in diameter. So human cells are about 100 times bigger than a bacterial cell. And incidentally, if you're wondering what's the problem with bacteria in our body, so bacteria are living cells. All living cells need food. So when the bacteria is in your body, it's looking for food. So what does it use for food if the bacteria is inside you? It uses you as food. It's a parasite, and it starts feeding on you. And it's eating your cells up. That's what it's doing. And they are multiplying in the process. All right, let's also show you one more picture of a prokaryotic cell, a prokaryotic versus a eukaryotic cell. For those of you who have the lecture outline, let's take a look at page B6. And on page B6, so on the top of B6, this is a bacterial cell, a prokaryotic cell. And basically what we see is it has a cell membrane or plasma membrane, but also surrounding the cell membrane is an outer cell wall, a bacterial cell wall. And this outer cell wall is always made up of a complex polysaccharide. Now, if you've had biology, you understand what the word polysaccharide is, hopefully. If you haven't had biology or didn't learn biology well, you probably don't know what a polysaccharide is. Poly means many, multiple. Saccharide means sugar. It's many sugars joined together. It's more complicated than that, as you'll learn in a microbiology class, for those of you who need that. But for our purposes at the moment, it's a polysaccharide that forms the outer cell wall. Inside the bacterial cell is this jelly-like cytoplasm or protoplasm. And cytoplasm or protoplasm in all cells, including human cells, is about 80% water. And after water, the next major chemical that makes up cytoplasm by weight are proteins. So it's really mostly water and proteins. Anything that's mostly made up of water and proteins has the consistency of Jell-O, because that's really what Jell-O is. So if you could feel cytoplasm in your hand, it would feel just like Jell-O. Now, inside the bacterial cell is a circular or ring-shaped chromosome made up of DNA. That's not a nucleus. That's not a membranous sac. It is simply a circular or ring-shaped piece of DNA or chromosome. So that's basically a bacterial or prokaryotic cell. Now, shown right below is a eukaryotic cell. Now, these are not drawn to scale, because if they were, this eukaryotic cell would be 100 times bigger. But this shows a cell with a nucleus, and not only does it possess a nucleus, it has many, many other internal little organs called organelles, as we wrote right here, organelles, little organs. And these are much larger cells, much more complex cells, and our human cells basically look like this, although they come in very, very different shapes. Now, returning back to page B1, just for a moment, so now what we want to do on page B1 is we want to start to talk about the parts of a eukaryotic human cell. And we're going to start with the cell membrane. Now, the cell membrane of any cell, including a human cell, creates a shape or boundary for the cell. And a term that you learned in biology is that these cell membranes are said to be semi-permeable. That means that some chemicals can get through the cell membrane and other chemicals cannot, whether they can get through or not. They're said to be semi-permeable. Now, the cell membrane itself is chemically made up of a double layer, a bilayer, two layers of phospholipid molecules with embedded proteins. And so we want to remind you of the structure of a cell membrane, which you learned in biology. So let's, again, return back to page B6. So back on page B6, all right, so on this page, B6 at the bottom, first of all, what I've drawn right here is a cell, all right? And it's got a nucleus. This is a eukaryotic cell, a human cell. And here's the cell membrane that forms the boundary or the shape of the cell. And of course, we've said that the cytoplasm, this jelly-like cytoplasm, is 80% water. It's mostly water. Let's just enlarge that for a moment. So it's about 80% water. And the next major thing after water are proteins. Now, something else you should know. All living cells have to live surrounded by fluid. There has to be fluid surrounding all cells or they will die. So surrounding every living human cell is fluid. We'll call that fluid tissue fluid. Now, if what I just said is true, which it is, you might say, wait a second, Professor Fink, hold on. You say all living cells, including human cells, have to be surrounded by fluid? Tissue fluid? Yes. Well, wait a second. My skin. Isn't my skin made up of cells? Yes, it is. Well, I don't feel any fluid. You're right. But what can we say about these cells on the surface of your skin? They're dead. Because they die when they're in contact with air. These are all dead cells on the surface of your body. If you've ever scraped your knee, scraped your arm, you may have scraped off some of these top dead layers of cells and seen a clear fluid start to ooze. That means you've scraped off the dead cells and you've reached the layer of living cells. That fluid that's oozing is tissue fluid that has to surround all living cells. If you were to make an incision in the abdomen and spread wide open the body, you'd see all this fluid surrounding all the organs of the body that's bathing those cells and keeping those cells alive with the nutrients and so on. So all living cells have to be surrounded by fluid. Now, what we'd like to do at this point is enlarge this area right here. We want to see this, this boundary, this interface between where the cytoplasm and the cell membrane and the tissue fluid all meet. And that's what this picture shows right here. This is simply the cell membrane enlarged, shown diagrammatically. So this is the inside of the cell, or cytoplasmic fluid up here, mostly made out of water. This is the cell membrane, and surrounding this is the outer tissue fluid, Now, the cell membrane is mostly made up of a double layer of phospholipid molecules. Now, you learned in a biology class about phospholipids. What does lipid mean? Fat. So commonly, these phospholipids are represented kind of like this. And when I see a picture of a phospholipid, it kind of looks to me like a balloon with two strings attached to it. Looks like a balloon with two strings. Now, I'm not going to try to make this class take the place of a college biology class. So I'm not going to get into the chemistry of all this. Well, you should have learned that in biology because you're going to need it for micro and physiology, which are much more chemistry intensive than anatomy. But the phosphate part of the molecule, the part containing phosphate, is this balloon. And phosphate, PO4, is said to be hydrophilic. Hydro means water, and philic means it likes. It likes water. These two strings are fatty acids. They are fatty acids, fats. And fats don't mix with water, right? If you have vegetable oil, which is a fat, and you mix water with it, they separate. Oil and water won't mix. So these fats are said to be hydrophobic. Hydro means water, phobic means hate. They hate water. So this is kind of a schizophrenic molecule. The balloon part is attracted to water, and the two strings hate water. Now, notice now the cell membrane. There are two layers of phospholipids. Here they are. And you'll notice in the inner layer of phospholipids, the balloon part that likes water is facing the cytoplasmic fluid on the inside of the cell. The balloon part that likes water, that's hydrophilic, is facing all the water on the inside of the cell. You'll notice that in the outer layer of phospholipids, the balloon part is facing outwards towards the tissue fluid on the outside of the cell. So the balloon part always orients itself towards where there's water. And you'll notice here in the middle is a double layer of fat. The way I kind of think of this is it's like an Oreo cookie. If you think of this as a cookie, and this is a cookie, this is the creamy filling in the middle. It's all fat. That's the creamy filling. Now, the purpose of the cell membrane, therefore, is it separates two fluid compartments. It separates the intracellular fluid, the cytoplasmic fluid on the inside of the cell, is separated from the outer tissue fluid on the outside of the cell by this double layer of fat, because water and fat won't mix. So literally, the purpose of this phospholipid bilayer is to separate this cytoplasmic fluid on the inside of the cell from the tissue fluid on the outside of the cell. Okay, now that we've reviewed what the phospholipids are for, what's all this stuff? Embedded between the phospholipids are various proteins. And some of these proteins that are embedded in the cell membrane, some of these proteins are located facing the cytoplasmic fluid on the inside of the cell. Some of these proteins are sticking out onto the outer surface of the cell membrane facing the fluid on the outside of the cell. Some of these proteins extend the full thickness of the cell membrane. Now, these proteins play very important functional roles that you should have learned in a biology class. I'm going to mention a few of their roles. You will hear much more about these functions, especially in a physiology class. Let's start with this protein right here. This protein is kind of shaped like a tube. This is like a cutaway view. It's like a pipe or a tube. And we could imagine, because this protein extends the full thickness of the cell membrane, we could imagine how certain chemicals might be able to flow right through this tube-shaped protein into the cell, or flow right through this tube-shaped protein out of the cell. These proteins are called ion channels. They're called an ion channel. And so they act like a tube, like a channel, like a pore, like a passageway for ions to flow through. Now, you should have learned in a biology class what an ion is. Ions are electrically charged atoms. Atoms are really small, and these are just electrically charged atoms, and they include sodium ions, potassium ions, calcium ions, and chloride ions. Now, in fact, these channels can open or close. And you're going to hear a lot more about that in a physiology course, for those of you who need to take that, which is most of you. So these ion channels can open or close, allowing these ions to either start to flow in or out of the cell. I should also mention that there are different ion channels. In other words, there are different separate sodium ion channels and separate potassium ion channels and separate calcium ion channels. So it's not like the same ion channel allows all types of ions to flow through it. They are very specific or selective as far as which ions flow through them. Now, this is all, whether it sounds very abstract. Some of you might be thinking, my gosh, I want to be a nurse. These biology teachers think that it's all academic and it has nothing to do with, you're wrong. If you've ever heard of a class of medications called calcium ion channel blockers, has anybody ever heard of that?
Yes.
Calcium ion channel blockers, which are used in the management and treatment of both angina pectoris, problems of the heart, and high blood pressure, among other things. The way these calcium ion channel blockers work is by temporarily blocking the calcium ion channels. That's a big subject that you'll hear more about in physiology. After all, what are drugs? Chemicals. And all these chemicals or drugs are acting at the cellular and molecular level. That's how they work. So there's no way that, I'm not going to go beyond this, but I can't emphasize enough how all of this cellular and molecular stuff is extremely important medically. All right, so that's ion channels. So they simply allow these really tiny ions to flow in or out of the cell. And you might say, well, so why are they important? These ions, or electrolytes, as they're also known, determine the electrical properties of our body. So the electrical activity of our heart, the electrical activity of our nervous system is all related to these electrolytes or ions flowing in or out of cells. That's all I'm going to say about that in terms of this anatomy class. Now, we have said that chemicals, nutrients like sugars, have to be able to get into the cell across the cell membrane. But sugars are too large to go through these ion channels. These are just big enough for these ions, these electrically charged atoms. So the way that larger molecules get across the cell membrane is by means of transporter or carrier proteins. There are proteins embedded in the cell membrane where sugars and amino acids can attach to these proteins, and these proteins then transport these sugars and amino acids across the cell membrane to keep the cell alive. So these transporter proteins or carrier proteins are what move or transport larger organic molecules. You learn in a biology class that a sugar like glucose, C6H12O6, is 24 atoms big. So it's 24 times bigger than that sodium ion. So it can't go through an ion channel, it has to be helped across. Sometimes some of these chemicals, the transporter protein in some cases requires energy in the form of ATP. And if it does, that's called active transport. And sometimes the transporter protein doesn't require ATP, and that's called passive transport. So, or facilitated diffusion. Yes?
So, like, you know how the glucose goes into the cell, but like, I know that there's, I forget how, but I forget you give insulin and something else to bring the sugar out of the cell. So how does it transport out?
How do you get sugar out of the cell?
Yeah, because I think it was like, you know, like.
Well, in the case of diabetes, you're trying to get the sugar to move from the bloodstream into the cells. And that's, insulin is a hormone. We're going to talk about this a little bit. Mostly a subject for physiology. Insulin actually is a hormone that activates this transporter protein. And by activating it, the insulin causes the transporter protein to start moving sugars into the cells. And as the sugar flows from the bloodstream into the cells, it lowers the amount of sugar in the bloodstream.
Okay.
That's how insulin works.
Okay, yeah.
It's going into the cell.
Okay.
The problem with diabetes is there's too much sugar in the bloodstream outside the cells, and it has to go into the cells. Some of these proteins in the cell membrane act as enzymes. That's a term you should have learned in a biology class. Enzymes are chemically proteins that catalyze biochemical reactions. Catalyze means they cause biochemical reactions to occur. Now, as I review this, since you don't, obviously this is all in your textbook, Chapter 2. Since I didn't ask you to buy the textbook to save money, that means that if you don't know this stuff already, you need to go and use those resources on my website where it reviews all this stuff. So you want to spend a lot of time on this. Some of this is covered in the lab manual, but some of this isn't. Obviously, a lab manual is not as thick as a 1,000-page textbook, so it must be missing something. All right, so you should know what enzymes do. They catalyze chemical reactions. Some of the enzymes may be facing the cytoplasmic fluid on the inside of the cell. Some of the enzymes may be facing the outer surface. Incidentally, when I say that a protein is facing the outer surface, if this is the cell membrane, if the proteins are kind of sticking out on the outer surface of the cell, they kind of are sticking out like that. So now another role of these proteins, very important, fascinating role, is that some of these proteins are sticking out on the outer surface of the cell membrane, and they act as receptor sites. Now, what are receptor sites? Receptor sites are where hormones, neurotransmitters, and drugs can attach. So hormones can attach to these proteins sticking out on the outer surface of the cell membrane. And when the hormone attaches to this protein receptor site, it activates it. And that's how hormones cause changes to start to occur inside of a cell. So, but again, I want to mention, so again, in terms of the picture, these could be very well receptor site proteins sticking out on the outer surface of the cell. Now, when I say hormones activate them, these protein receptor sites are specific for certain specific hormones. In other words, there are insulin receptor sites sticking out on the outer surface. There are growth hormone receptor sites. There are estrogen receptor sites. So they are very specific. So, well, like a puzzle. So, in other words, some cells might have just one or two types of receptor sites on their outer surface, and therefore they're only affected by one or two hormones. And other cells of our body might have 20 different receptor sites on the outer surface, so 20 different hormones can affect that cell. This helps explain how it's possible that some hormones only affect certain cells of our body. For example, estrogen obviously affects the cells of a woman's uterus. It affects these uterine cells, but it doesn't affect heart cells because heart cells don't have estrogen receptor sites. Interestingly, in terms of insulin, almost all cells do have insulin receptor sites. So again, cells can vary as far as which receptor sites to which hormones they possess. Now, we said that not only are there receptor sites for hormones, there are receptor sites for neurotransmitters. You'd say, what are those? Neurotransmitters are chemicals released by nerve cells. So neurons can release chemicals. These are known as neurotransmitters. So if insulin and growth hormone and estrogen are names that you're familiar with as hormones, some examples of neurotransmitters would be serotonin, norepinephrine, and dopamine. So if you've heard of dopamine and norepinephrine, acetylcholine, serotonin, those are the names of neurotransmitter chemicals. And again, these chemicals attach and activate specific receptor sites on certain cells. Again, not all cells have all these receptor sites. Some do and some don't. So again, this helps answer the question. Let me just say one more thing. In addition, this is where drugs can attach. So drugs are designed to attach and activate certain receptor sites. So this helps explain that when you take a medicine, you take a pill, over the years students would sometimes ask me, well, how does the medicine know where to go? And the answer is, it's absorbed into your bloodstream and it goes everywhere. But it can only affect those cells of your body where there's a receptor site where that drug can attach. So if you're taking a medication for your heart, that medication has been designed to attach to receptor sites on heart cells and not on other cells. However, sometimes, even though they design a drug to activate receptor sites on the heart cells, there may also be receptor sites on other cells of the body where that drug also attaches and activates. That's what creates side effects. Side effects of the drug is because that drug not only activates the receptor sites on the location where we want it to act, but it's also activating receptor sites in other places of the body where we didn't want it to act. An example of that is when you take an antihistamine. Antihistamines are drugs where we want them to act on certain cells that are associated with when you have an inflammation, you have a cold, and so on. But antihistamine drugs also activate receptor sites in your brain, causing you to get sleepy. So that's, most antihistamines cause drowsiness because these same drugs that affect our nose when we've got a cold also activate and affect brain cells, tending to cause us to get sleepy. That's a side effect. Now, another role of these protein receptor sites, these proteins in the cell membrane, is some of them act as recognition sites. Now, these recognition sites are known as glycoproteins. So, what's a glycoprotein? Again, these are always facing the outer surface of the cell, and a glycoprotein is a protein with sugars attached to it. And glyco, like the word glucose, means sugar. So these are sugars or carbohydrates. Carbohydrates are sugars. So when you have carbohydrates or sugars attached to a protein, that's called a glycoprotein. That's what these recognition sites are. Now again, they are always sticking out on the outer surface of the cell membrane. Now what are they for? I wrote that these recognition site glycoproteins allow your WBCs, your white blood cells, to recognize your cells and distinguish them from foreign cells. You'd say, what? All right. Every single person, every one of us, has a unique set of glycoprotein recognition sites on the outer surface of every cell in our body. So there is a combination of different recognition sites on the outer surface of the cell membranes of every single cell in your body. Let's imagine that a bacteria enters your body. Your white blood cells are kind of cruising through your body looking for suspicious characters. Now, how do your white blood cells know when they bump into a cell? How do they know if that cell they bumped into is your cell or a foreign cell? All your cells have the same recognition sites on their outer surface. Any cell that enters your body that doesn't have the correct recognition sites, that white blood cell will destroy. It will swallow up and destroy. So when the bacteria enters your cell, it doesn't have the correct recognition sites on its outer surface, and your white blood cell proceeds to destroy it. Now this is not only true that your white blood cells will destroy bacteria that enter your body, it would also be true in what happens when they try to do an organ transplant. Because the cells of every single one of us have a unique set of glycoprotein recognition sites. The only people who have the same recognition sites are identical twins, because they're identical. But if somebody doesn't have an identical twin, which I assume is probably all of us, then there's not a single person in this entire universe that has the exact same recognition sites on their outer surface. So if they were to take a kidney from somebody else and stick it into you, or vice versa, the white blood cells are going to bump into those transplanted kidney made up of these foreign cells. They're going to say, these are not our cells. They don't have the correct recognition site. And your white blood cells, or immune response, this is your immune response, those white blood cells are going to start to destroy the cells of that transplanted kidney. This is known as organ transplant rejection. I'll just write the term. Organ transplant rejection. So organ transplant rejection is simply when your white blood cells start to destroy those foreign cells. Even though they're human cells, they're not the same as your human cells. So one of the things, yeah, I'll say this, so one of the things you've heard is before they try to do an organ transplant, you've heard the expression, they're looking for a close match. They're looking for, they identify the recognition sites, the type of recognition sites on the surface of these cells, And this, incidentally, this idea of recognition sites is the same idea as blood type. You know how some people are blood type A, some are blood type O, some are blood type B, and you have to have the right blood type? That's the same idea. It's these recognition sites. Those are also recognition sites on red blood cells. So they're looking for a close match. If they can't find a close match, they won't even attempt to do the transplant. If they can find a close match, they will do the transplant of the kidney, of the heart, of the liver, whatever they're trying to transplant. But even then, if it's not a perfect match, which it won't be, they have to somehow suppress the immune response and the white blood cells from destroying that transplanted organ. And that's why every person who has an organ transplant is placed on immunosuppressant drugs. Now, what does the term immunosuppressant mean? To suppress or inhibit the immune response. And the way they work is they lower the white blood cell count. They lower the white blood cell count. So if you know of anybody who has had a kidney transplant, they are taking immunosuppressant drugs. The most commonly used are corticosteroids like prednisone. And what it does is it lowers the white blood cell count. What they're trying to do is to lower the person's white blood cell count, so to try to slow down the white blood cells, the immune system, from attacking and destroying this transplanted organ. Now, the problem is obvious. If you put somebody on a medication so they can tolerate this transplanted kidney, if you've given them a medication that weakens their immune response, what have you made them more vulnerable, more susceptible to? Infections, viruses, bacteria. You're hindering their immune response. But that's the trade-off. Basically, the doctors arrive at the following analysis. They say, well, you need a kidney or you're going to die. You need a heart or you're going to die. If we can find a close match, we recommend doing the transplant, and even though we're going to put you for the rest of your life on immunosuppressant drugs to prevent your own body from rejecting that organ, if necessary, we'll have to put you on antibiotics all the time as well. All right? And this is the complications of medicine. This is why sometimes when they try to solve one problem, they create other problems in the process. The person had a kidney problem, they put a transplanted kidney, they solved that problem, and they've created a new problem of increased vulnerability and susceptibility to infection. So it's not unusual that somebody who gets a kidney transplant ends up dying from something like pneumonia. But if you didn't give them the kidney transplant, they would have died at even a sooner time from kidney failure. Yeah?
With something like that, taking the antibiotics, how would that help if they're taking...
Well, the antibiotic kills bacteria. The problem is antibiotics don't do anything against viruses. All right? The antibiotic has nothing to do with the immune system. It kills bacteria.
Virus, your body would have a harder time.
That absolutely, and that's why somebody would commonly die from viral pneumonia. Because antibiotics don't work and help against the viruses. Yeah?
But then with the prednisone, that builds it back up, though. Is that correct? Because it builds up the hormone?
No.
Steroids?
No, it is used as a hormone, but in this case it's suppressing the immune system. It has multiple actions. Wait until you have to start memorizing all the things that drugs do. Coming attraction. All right, so one last comment I want to make about these recognition sites. And I wrote the term right here. Autoimmune disease. Now, some of you have heard that term, autoimmune. What does that literally mean? It literally means you're immune to yourself. You'd say, what do you mean I'm immune to myself? Well, in other words, some people's white blood cells attack their own body, their own cells. Well, obviously, the white blood cells are designed to attack foreign cells and not attack your own cells. But sometimes this whole mechanism breaks down. It doesn't work right. So your very same white blood cells that are supposed to protect you against bad guys start attacking the person's own cells. Now, that's called autoimmune, immune to yourself. There are hundreds of different types of autoimmune diseases depending upon which part of the body the person's white blood cells are attacking. Just to name a few examples. If your white blood cells or immune system attacks the joints of your body, that's called rheumatoid arthritis. If your white blood cells attack and destroy your pancreatic islets, that's called type 1 diabetes. Type 1 juvenile onset diabetes that develops in childhood is an autoimmune disease. The person's own white blood cells attacked and destroyed the gland that produces insulin. That's why they have to inject insulin for the rest of their life. Rheumatic heart disease is when the white blood cells attack and destroy the valves of the heart. Multiple sclerosis, MS, is when the white blood cells and immune system attack your nervous system. There are hundreds of different types of autoimmune disease. Ulcerative colitis, the immune system attacks the large intestine. Crohn's disease, it attacks the small intestine. So there are hundreds of these. Graves' disease, the immune system, the antibodies, the white blood cells attack the thyroid gland. So there are many, many types of autoimmune diseases. Obviously, the breakdown happened. Either the white blood cells aren't working right because they're not recognizing these recognition sites, or for some reason, and this is more likely, certain cells of the body are lacking those recognition sites. They don't have the markers to identify those cells as being your cells. So the white blood cells don't see them as your cells, they see them as foreign cells. So you'd say, well, why would that happen? In almost every clinical disorder, there is almost always some sort of genetic factor. There's a genetic factor in almost every problem. There may be other factors in addition, but there's genetics usually behind this. So in other words, autoimmune diseases do tend to run in families. All right, so we have talked about, about 5 different types of proteins in the cell membrane. We've spoken of ion channels, talked about transporter proteins, spoken of enzymes, talked about receptor site proteins, and then we've spoken of recognition site glycoproteins. If we return back to page B1, so back on page B1, letter D, we wrote that each person has a unique set of glycoproteins on the surface of their cells. Those are those recognition sites that allows your immune system, your white blood cells, to recognize which cells belong to you and which cells are foreign. So let's just review with you real quickly what we said. We said that the ion channels are specific for specific ions. So in other words, one single ion channel does not allow any and all ions to flow through it. They are specific. So there are specific sodium ion channels. I'm sure you have this in your notes already. There are specific potassium ion channels, chloride ion channels. And these ion channels can open or close. The transporter or carrier proteins are also specific. Some of these proteins help transport sugars, like glucose, across the cell membrane. Other transporter proteins transport amino acids across the cell membrane. In some of these cases, energy is required in the form of that high-energy nucleotide, that gasoline that powers everything that occurs in the living cells, called ATP. And when energy in the form of ATP is required, that's called active transport. Sometimes it doesn't require ATP. In that case, it's known as passive transport. Of course, the role of enzymes. Enzymes are very important in living things, all living things. Enzymes are proteins that catalyze chemical reactions, I should say. All right, so we talked about all this last time. We also mentioned receptor sites. We said receptor sites are always located on the outer surface of the cell membrane. They are specific, again, for certain hormones, certain neurotransmitters and drugs, so that the insulin activates certain receptor sites. Growth hormone activates different receptor sites. Estrogen activates different receptor sites. And not every cell possesses receptor sites for every hormone. Some Some cells have receptor sites to just one or two hormones. Some have receptor sites for 10, 12, 15 different hormones. And that pretty much explains why hormones are selective and specific as far as which cells they affect based on the receptor sites that are on them. And of course, the activation of the receptor site leads to changes in the cellular activity. And then the fifth thing we spoke of last time are recognition sites. These are glycoproteins that are located on the outer surface of the cell. And they're called recognition sites because they allow your white blood cells to recognize which cells are yours and which cells are foreign. And so when any foreign cells enter our body, they lack the correct recognition sites on the outer surface, so the white blood cells proceed to destroy them. All right, so that's a quick review of everything we talked about previously. Now, in letter E, I wrote that some cell membranes are folded into microvilli to increase the surface area. You might say, what? Cells in your body come in different shapes. They're not all perfectly spherical or round. Some cells in our body are cube-shaped, some are tall columnar-shaped, some can change shape, like white blood cells. But looking at these two cells, this is what a typical stomach cell looks like, the cells lining the inside of your stomach. And this is what a typical cell lining the inside of your intestines looks like. Now you'll notice that both cells kind of look similar. They're kind of tall, rectangular shaped cells. But you'll notice that the cells of the intestine the cell membrane, at least on one side of the cell, is folded, where the cell membrane is folded into these finger-like projections that are called microvilli. Now, the word villi means finger-like. Make sure you learn that. That root, villi, is going to keep reappearing. It means like fingers. Microvilli means microscopic fingers. So the cell membrane is folded into what look like little fingers sticking out. Now, notice that the cell membrane of the stomach cell is straight. It doesn't have those finger-like extensions. Now, just a little bit earlier today, I was telling you about the surface area of a cell, right? And we said that the surface area is how nutrients get across the cell membrane into the cell. So let me ask you this question, back on this picture. If this cell membrane is folded and it goes in and out and in and out and in and out, which is what a hamburger is all about, but no, that's something else. All right, but because the cell membrane goes in and out, can everybody see that that's a much bigger surface area?
Yeah.
Because if we stretched it out, it would be much bigger than if it was just a plain flat cell membrane. So this has a larger surface area for nutrients to go into the cell. And so we see that the cells of the inside lining of our intestine are designed to increase absorption of nutrients. They have a larger surface area to facilitate and make it easier for nutrients to get into the cell for absorption. On the other hand, the stomach cell is not designed that way. It's just a flat cell membrane. It doesn't have this greater surface area. So in fact, very few nutrients are absorbed from your stomach into your body. There is very little absorption of food, of nutrients, across your stomach. Almost all the absorption of nutrients is absorbed across your intestine. So literally, the structure reflects function. The reason why the cells of the intestine are better at absorbing nutrients than the stomach cells is reflected by the difference in structure. So that's the purpose of the greater surface area.